Hello, and thanks for joining us today for the latest Technology Networks webinar. How can metabolomics accelerate natural product discovery? I'm Rory, a science writer for Technology Networks, and I'm here to moderate the event. Very excited today to be joined by Roger Linnington, who will be presenting today's webinar. A very warm welcome to Roger. Roger is a professor in the Department of Chemistry at Simon Fraser University in British Columbia. Roger's career has seen him conduct research across the globe, beginning with an undergraduate degree at the University of Leeds in the UK, before moving to Canada, where he received his PhD from the University of British Columbia. Subsequent work in Panama and at the University of California, Santa Cruz has seen his research formally recognized with awards such as the American Society of Pharmacognosy Matt Suffness Young Investigator Award. Today, Roger will be speaking about his group's work in building and maintaining the Natural Products, or NP, Atlas, a world-renowned open access database that lets researchers view the chemical diversity of the natural world. Following the webinar, we'll have a Q&A session with Roger. We would welcome any questions that you may have. To ask a question, you should enter it into the box on the left-hand side of your screen and click Submit. And we're going to try and answer as many as we can during the time we have available. If you experience any technical issues, please click the question mark in the top right-hand corner to request support. We'll now begin the webinar. Over to you, Roger.
Welcome to the next in the Waters webinar seminar series. My name is Roger Linnington. I'm an associate professor in the Department of Chemistry at Simon Fraser University. And today I'm going to be telling you about a recently released open access data platform called the Natural Products Atlas, and how the Atlas can inform and advance natural products discovery workflows. So the principal objectives of the webinar today are to introduce the Atlas platform itself, to describe its scope and its main features, and then to explain how this data set can be integrated in untargeted metabolomics programs, and to illustrate how some of the advanced features in the Atlas can advance those discovery models and provide additional value and opportunity. So if we think broadly about how natural products metabolomics workflows proceed, they normally start with a set of source organisms. So in our laboratory, these are microbial natural products libraries, but these could equally well be plant-derived or marine invertebrates. And these samples are subjected to an in-house extraction and sample preparation workflow prior to analysis by LC-MS. So in our laboratory, we use the IonAbility QTOF, but again, this can be substituted by any number of different instruments. And these raw data are then processed using either open access or commercial software tools. And in the Waters environment, those options are either Progenesis or Unify. And these can be optionally incorporated into external databases like the Natural Products Atlas in order to annotate and describe the sort of chemical landscape of the sample set being analyzed. So the principal goals of today's webinar are to explain how the Atlas can improve identification strategies in some of these data analysis pipelines, but also to demonstrate how tools that are available in the Atlas infrastructure can allow you to further refine and improve identification strategies. And so we're going to look first at the Atlas in general, then how it applies to natural products metabolomics, and then how we pursue this refinement strategy. So the first stage then is to talk about the Atlas and how it was built and what its mandate is. So the Natural Product Atlas is a completely free resource. It's open access, meaning that it is fully downloadable. The entire data set is available for free and is provided under a Creative Commons license that allows anybody to reuse it for either academic or industrial purposes. The long-term objective is for this database to contain all the known microbial natural products, and that's an ongoing effort that we'll discuss in a few slides' time. The platform is fully searchable using a variety of different search tools. It also incorporates tools for chemical diversity visualization so that we can describe not only individual compounds, but also how those compounds fit into the landscape of chemistry for microbial natural products. Because it's an online resource, it works on any operating system, including PC and Mac. And it's also designed to work on mobile platforms, including iPads, tablets, and even phones as well. So if we step back and think about how the atlas was originally conceived, we originally designed a strategy to create a central, open, community-supported repository for data on all naturally occurring microbial natural products. So this was our sort of mandate at the beginning. And we aimed to include data from both the primary literature and patents to find and incorporate all structures, compound names, and appropriate synonyms, to abstract representative physicochemical data, to incorporate bioactivity data, to list all the instances of total synthesis for all of these molecules, to find and incorporate information on all the structural reassignments, to incorporate the taxonomy for the original producing organism, and ultimately to produce a full list of taxonomy for all the producing organisms from which each compound has been found. Obviously, this is a very ambitious set of objectives, and this is a set of goals that we are working towards. So currently, the atlas incorporates those components which I've highlighted here in green. So we have the primary literature, some information about structures and compound names, taxonomy, and then the sections that are in orange are either in the process of being incorporated or we are designing strategies to be able to incorporate them. So the development of the atlas is a sort of long-term project, and these things in orange are some of the next things that we aim to try and address. So if we look at the contents of the atlas, currently the database contains a little over 25,000 compounds, and we are performing an ongoing data curation effort to capture not only the data published now in the most recent journal articles, but also to look retrospectively through the literature and find other molecules which have been omitted from the database for one reason or another. So this is a sort of, this backfilling effort is an ongoing work, and we have about 5,000 compounds in hand which will be incorporated into the next database release later in the year. So if we look at those compounds are distributed, a little under 10,000 of them are bacterial natural products, while about 15,000 are derived from fungal sources. And if you look in the bottom left here at the taxonomic distribution, if we look at the whole data set, we find that about a quarter of all the molecules in the data set are derived from Streptomyces, and a little under another quarter derive either from Aspergillus or Penicillium, which are two fungal genera. The remainder are scattered sort of throughout the taxonomic tree, you can use these data in a variety of ways, as we're going to see later on. In the bottom right, we see the distribution of compounds based on year of discovery. And as you might expect, in the early years of this trace, the number of compounds being found per annum is very low. So this rate of discovery remains low, slowly increasing from the 1950s through to the end of the 1980s, at which point the advent of two-dimensional NMR techniques, as well as the improvements in mass spectrometry, led to a sort of acceleration in natural products structural elucidation. So you can see in the early 90s, the rate of discovery really picks up. And when we reach modern times, somewhere in the order of 1,400 molecules a year are being discovered. So this is an ongoing effort on our part to keep this database up to date with these new structures. A reasonable question one might ask is why the atlas is needed at all. And in fact, there is a very nice review which has just come out in the Journal of Chemoinformatics by Sorokina and Steinbeck, where they describe no less than 122 resources for natural product structures developed since the year 2000. So this is a remarkable cross-section of databases, and it might seem that the database landscape is already full. But when you look more closely at the contents of those databases, the situation is not quite as promising as it first looks. So there are a number of very nice commercial tools. So a few examples are illustrated here: Dictionary of Natural Products, Marinlit. But these databases are inherently sort of closed-ended. You cannot download all the structures or export all the information to third-party tools. You can't insert them as scientific libraries in data processing software of your own, which really limits them to sort of one-by-one queries based on individual structures. There are also some academic database efforts. So NPAS, StreptomeDB, Supernatural2, these are just 3 examples. But these either have a limited scope, so there are a number of databases which are focused on particular regions of the world or particular subclasses of organisms. StreptomeDB is a great example. It's a terrific database, but focused exclusively on Streptomyces, so it doesn't include chemistry from other bacterial sources. Supernatural2 has many structures from lots of different sources, but is not downloadable as a complete data set either. So really there was no free comprehensive microbial natural product database, and this is what motivated us to pursue this project. So if we look at how the Natural Products Atlas is structured, we have 2 components. There is a database backend, which is where we store and manage all of the information about each compound. And then there is a web-based front end, which allows users to query and explore the data in different ways. And that front end is split into 3 principal sections, which we term search, explore, and discover. So the search region provides several different ways in which you can search the data set for compounds and compound classes. The explore section is the section which provides different visualizations for chemical diversity at different levels of granularity. And the discover section provides a growing suite of dashboards which allow you to search the database from alternative viewpoints, such as authors and other data sets like this. And we will see examples of all of these here in the next few slides. So if we start with the search component, if we go to the front page of the website and we go to the search drop-down bar here, you'll see that there are 2 choices: basic search and advanced search. We're going to look at basic search now, and advanced search will come up later in the talk. So if we look at the basic search page, we see that the basic search interface includes a structure drawing plugin, which allows you to enter either complete structures or substructures or even sets of substructures, and to search either for an exact match using the full structure radio button here, or substructures, or alternatively similar molecules using this similarity threshold, which can be very helpful for finding analogs and other related compounds. Alternatively, you can also include compound name, formula, accurate mass information, or even information about taxonomy in your search terms. And you can do this with or without the structure component. Once you complete a search, you get a results table below this page. And if you follow any of those results to the corresponding compound page, this takes you into the explore component of the website. So the explore component, you can see here in the drop-down menu, explore component includes 4 different levels of granularity. So in the first level, at compounds, you're going to see information about individual molecules, and we'll look at that in just a sec. Clusters are groups of molecules which share high structural similarity to one another. Nodes are groups of clusters which share some similarity, but not the same high degree of similarity to one another. And then global is a visualization which shows you the entire chemical space landscape of the whole data set. So this is really akin to sort of Google Maps view of chemical space, where compounds would be akin to houses, clusters might be similar to towns, nodes would be provinces or states, and then global would be your sort of Google Earth view of the whole thing. So if we look at what data are available in each of these cases, so here you can see on the right-hand side, the compound page for one of the molecules in the data set. There's a picture of the structure itself. There are some relevant pieces of data about the molecule on the right-hand side, including its taxonomic origin. There are structure representations, which you can copy and paste into ChemDraw or other search tools of choice. There are a number of export options down here, so you can export the structure as an image with the PNG. You can export the structure as a mol file, which you can open in ChemDraw or other drawing programs, or you can export all the data on the page as a tab separated value file, which you can open in Excel or other data formats. Below this, there are also sometimes external links to other databases. So we've put significant effort into connecting the Natural Products Atlas with other natural products centric data sources. So in this particular case, there is a link to MIBIG, which is the database of biosynthetic gene clusters run by Marnix Medema in Wageningen University in the Netherlands. And there's also a link to GMPS, which is the MSMS experimental natural products data set run by Peter Dorstein and his team at the University of California in San Diego. We're going to come back to these external links later on. They're really quite powerful tools for natural products discovery. So you'll see here that each molecule also includes a cluster ID and a node ID. So the cluster ID takes you to the group of molecules which all show very strong structural similarity to your molecule of choice. So if we go now to clusters, what you'll see is that in this case, the molecule that here is a cluster demonstrating a set of molecules, and you'll see that actually they fall into two subgroups. There's a sort of northern half and a southern half, and there is very high interrelatedness between the two groups in the north and the south, but they share some separation to one another. You can hover over any one of these dots. So every dot is a compound, and if you hover over any dot, you'll see a pop-up window there which shows you something about its structure and also its identifying number. If we take another step back in terms of chemical space and we go to the node view, now you're looking at clusters of molecules which relate to one another. So the size of this hexagon tells you how many compounds are in that class. And this tells you something more general about how is this area of chemical space occupied by natural products which are in the data set. And then lastly, if you go all the way to global, this is it's actually a spherical representation of all of natural product space, which illustrates how nodes are related to one another in terms of structural features. And we're going to see, it's a little hard to see in this planar view, but we're going to see a short video which illustrates its shape a little bit better in a second here. So if we look at how you might use this, if we start in the compound page, compound search page, if you type a compound name of choice, the system will autofill and give you the options available to you. If you submit that, then it takes you to the comp page, and you'll see at the bottom here the original isolation reference and hyperlinks that take you to other sections of the page. So if we go to the cluster ID, this takes us then to the relevant cluster. The reference compound we started with is highlighted in red, and we can go from there one level back again to the node view. Again, the origin of the compound you started with is highlighted in red. Other compounds are similar but somewhat different. And then in the global view here now, we'll be able to see how you can manipulate this global sphere. So this tells you which other nodes are nearby, what kinds of classes of compounds might be considered structurally similar. So the last section of the Atlas infrastructure is the Discover section. And as I said, this includes dashboards which provide different views on the Atlas content. And the long-term goal is that this section will be augmented with additional dashboards as we receive requests from the community and as people come up with other ideas for visualizations that will be useful. So we're going to look at just one example today. That's the author dashboard. So it's possible to search the data set based on any author, provided that that author has an up-to-date profile in the ORCID author database. So you have to have created a profile there and associated your articles with your profile in ORCID, but provided you've done that, then the Atlas can search for the right articles very easily. So if you search by name, we're going to use as an example my close colleague Kerry McPhail from Oregon State University. And so if we put her name into the search tool, ORCID returns only one entry, which we can select. And this entry then provides a summary of all the data which have been found associated with this author. So you'll see here in the middle, it tells us how many articles were returned from the ORCID database, in this case 69 in total, and then how many of those are associated with the discovery of microbial natural products, in this case 13. So down below we see a pie chart which describes the journals, distribution of journals where these articles were published. And then below that we also see a timeline which shows which compounds were published in which years, as well as a sidebar at the bottom which shows you the structures that go with them. And then if we scroll even further down, we find also a table of each of those compounds with hyperlinks to each journal article, information about the citation, and also information about which compounds belong to each of those papers. So these are ways in which researchers can explore the information about natural products, chemical space in very broad ways. And we're going to see in a short while how we can use these then to inform the metabolomics analysis. One question that we get asked a lot is how the atlas was built and how we did this. So this was certainly not an individual effort. So although my laboratory maintains the atlas, we required the assistance of a huge group of international collaborators in order to make this project a reality. So this is it really is a community-driven effort built through the hard work of lots of people from all around the world. So current collaborators are listed on the right-hand side here. And we certainly appreciate their contributions a great deal. In terms of the mechanics of how the database was constructed, there isn't time to really go through all those details in this presentation, but we have built significant architecture in the background in order to allow us to do this efficiently. So what you're seeing here is a screen grab from our separate curation online database system. So this is a separate tool used only by curators in order to review, correct, and submit data from articles as they are identified by our text mining and article mining tools. So here you can see information about the article itself. So here in the top left, we have all the information about the title and the authors and all the other citation information. We have information about how many compounds are in the paper. On the right-hand side here, we have clickable tabs which show you the structures of all the new compounds that are going to be inserted, as well as their names and also which organism they came from. And so all this information is reviewed on an article-by-article basis by this group of curators and is then submitted and subject to final quality control checks before we insert it into the database. So if people are interested in learning a bit more about that, I would certainly be happy to talk to you about that offline. One of the questions which I'm sure audience members are most interested in is whether they can link the atlas to their own tools. And the answer to this is absolutely yes, you can. That was one of the sort of driving reasons for doing the atlas project in the first place. So there are two elements of the website which are important for this. The first is this download tab here. So if you go to the download page, you'll find a downloadable TSV file which contains all of the data available in the atlas for every compound. So it's available, it's a completely open flat file. And that file can be reworked and reconfigured in order to import it into any other external program as a scientific library. If you run web-based tools and you want to make link outs to pages in the atlas, if you go to the developer section here of the website, there is information and instructions on how best to do that. So there are a set of tools available for developers as well if you want to incorporate atlas data into your own project. So one of the advantages of having built such a data set is that it gives us the opportunity to look quite broadly at the sort of state of play of natural products discovery. And so what I'm going to do next is I'm going to show you just a little bit about how we might use this data set to learn something about the scope of natural products diversity and how learning about that scope assists us when we think about how we then pursue metabolomics characterization. So one of the first things that we did once we had this data set in hand is we looked at how broad the chemical diversity of the microbial natural products world really is. So an obvious question we might want to ask as mass spectrometrists is how redundant is the data set in terms of isobaric species? How often do you find the same formulae for different molecules over and over? And so in order to ask those kinds of questions, what I've done here is I've taken all the unique formulae in the Natural Products Atlas. I've counted how many times each unique formula appears in the data set. And then I've arrayed them based on increasing carbon number followed by increasing hydrogen number, and then all the other elements alphabetically as usual. And you learn a number of really important things straight off the bat. So clearly there are a few formulae which have very, very large numbers of isobaric species. So the highest numbers are over 100 isobaric components. But what you see quite quickly thereafter is that most of the other formulae don't have nearly that degree of redundancy. And in fact, the number of formulae with more than 20 isobaric species is already quite low. You see some general trends in this data set, which perhaps we would expect. So this tallest spike here, these are all the C15 containing molecules. The C20 containing molecules have a reasonable high density, not so much for C25, but there's another high density spike here at C30. And this makes sense based on what we know about biosynthetic origin of natural products and how natural products are put together. It's reasonable that these should have fairly high numbers. But if you look closely, you see an additional evidence of additional periodicity within the data set, which is also intriguing and perhaps at first glance a little bit unexpected. So what we're going to do is we're going to zoom in here and we'll take a look in a bit more closer detail at just the C20 containing molecules. So if we expand this plot just to contain those molecules with 20 carbons, then what we find is that there is indeed a periodicity to this, and this periodicity is driven by the constitution of the formula. So all of these large spikes which you see going across the data set here are caused by sets of formulae which contain 20 carbons and then either 16, 18, 20, etc. Protons, and then some variable number of oxygens between 1 and 5. And this is also reasonable if you think about the biosynthetic origins of natural products. So many polyketides, terpenes, carbohydrates, and other classes are made up exclusively of building blocks containing carbon, hydrogen, and oxygen. So it's reasonable that we should have a higher number of isobaric species in those cases. But one of the things to me that really stands out about this data set is that if your formula contains anything other than carbon, hydrogen, and oxygen, then in most cases, that formula is unique in the data set. And in some cases, there are 2 or perhaps 3 candidate compounds. But that provides a very different analytical challenge when considering de novo annotation. So for many of the formulae that are in this data set, the question is no longer which of these 25,000 molecules might match with my product, but rather, is this peak this particular molecule or not? And I think that realization and the information that we get from that can be sort of fruitfully deployed when we're thinking about how to annotate large, complicated, unknown sample sets. So we can also refactor these data in different ways. So if I take the same data set and now I refactor it so that we are counting the number of molecules associated with each formula, what we find is that as we know, you can't see all of the bars here because some of them are very small. They're sort of down in the baseline. But the most predominant formula has over 130 matching molecules. But most formulae in the whole data set have only 1 matching molecule, and many of the remainder have only 2. So if we zoom in on just this early part of the data set, so this is just an expansion of the first 10 data points, There are, at the moment, 11,500 total formulae in the database. 7,600 of those are unique, and a further 1,700 match to only 2 compounds. So close to 85% of all the formulae in the database are either unique or have a pair of matches, which I think is pretty useful information to know as we move forward with the annotation work. So another advantage of the atlas is that we have included the information about the taxonomy of the producing organism. And this also gives us an opportunity to look at the data set in an orthogonal, from an orthogonal perspective. So what I'm showing you here is an alternative viewpoint on chemical diversity within the atlas. So in this case, we've taken the cluster information from the structure database. So remember that clusters are formed by grouping together molecules which have very similar structural features. And I've then color-coded each of these clusters based on the type of organism from which they were first discovered. So in this case, bacterially derived natural products are colored in blue, and fungally derived natural products are colored in green. So if we zoom in and we look at just one region, so we're just going to go to this expansion, what we find is that, rather remarkably, the overlap of origin within a given cluster is generally very, very low, which means that there is comparatively little overlap in chemotypes between bacterial and fungal sources of natural products. Now, one caveat with this analysis is that the atlas only includes the taxonomic information about the original isolation. So it doesn't yet include the information about all the instances from which compounds have been discovered. So that's a project which would need to be done in the future. But even within that caveat, it's pretty clear that there is a very high degree of segregation between chemotypes that derive from bacterial sources and chemotypes that derive from fungal sources. And so these data demonstrate that it is reasonable to subdivide the library if you're searching against a sample set where you know the origin of the materials that you're studying. So if we take that same formula distribution and we now review that formula distribution based on either bacterial or fungal origin, we learn a number of really important things. So if we start with the bacterial plot, so again, this is counting the number of molecules which match with each formula. So 1 means there's only a single molecule in the database with the given formula, and so on. So in the bacterial only version, the highest number of matches is only 14, which is rather remarkable. That's not true in the fungal case. So this fungal plot has been trimmed in order to fit on a page like this. So in the fungal case, the maximum number is 120. So it appears that there is a much higher degree of formula redundancy in the fungal data set than there is in the bacterial data set. It's also true that at the moment, the number of instances where formulae are unique is higher in the bacterial case than it is in the fungal case. Although you have to be careful about that analysis because the total number of compounds is not the same in each of these 2 data sets. So you have to be a bit careful about that. Nevertheless, with the current data set that we have, this information shows that even the highest degree of isobaric overlap in the bacterial data set is now really quite low. And so that's also going to have a very positive impact on assignment strategies and methods for identifying unknown molecules. So what I would like to do now is talk a little bit about how we can integrate the atlas with that discovery workflow. So if we go back to this slide we introduced at the beginning, raw data can be acquired from any of a huge array of different instruments, and there are now lots and lots of different tools for doing analysis on those raw data sets. And many of these allow you to incorporate some kind of list of candidate structures against which to compare. So what I'm going to show you in the last slides is a demonstration of integration with some of the Waters software tools. But this could equally well be done with open source or vendor platforms from other places. So the Natural Product Atlas is an open resource, and researchers are free to use it in any platform that they choose. So we have refactored the Natural Products Atlas data output, and we have created scientific libraries which are directly importable into either of the two data analysis platforms available for Waters instruments, so both Progenesis and Unify. I'm going to use Unify as my example because it's the software platform I am more familiar with. So if we look at what happens in Unify when you import this scientific library, so you can download the scientific library as a single file. You can import it into Unify as a scientific library tool. And then what you get, so here we have an example page. So this is an example of a single molecule. You can see that much of the information that's in the atlas is presented here under various fields on the compound page. All of the compounds available are listed here on the left, and you can scroll through them and find the ones you want. You can search for them in various different ways. And then importantly, there are a number of properties in the box down here. So we're going to come back to this synonyms box a little bit later on. The scientific library has a number of item tags, which is pretty helpful because it allows you to filter the data in different ways. So if we go to the item tags pop-up, which is on the right, I'm just going to expand this region so we can see it a little bit better. So this pop-up allows you to select tags which you want to consider. So there are several core tags which are useful to this community. So at the bluntest level, there is an organism type tag which allows you to filter for only bacterial or only fungal compounds out of the data set. Alternatively, if you want to filter at the genus level, if you were to click this drop down, you would see a list of all the genera that are in the data set, and you can choose a genera of choice. So if you want to study particular groups, that's certainly very possible. You can even filter at the species level if you so choose. So what we did as a proof of concept is we took a sample from our data set. So we took a natural products extract from a microbial isolation, and we doped into it a set of 20 natural products of known identity in order to have a reference sample where some of the materials were unknown, but some of them were known, and we could test the effectiveness of the system at identifying those known metabolites. So if you take any particular unknown sample, you import it into Unify, you run the peak picking and other standard processing steps, and then you apply the scientific library against that file. You receive a component summary, which is shown here on the top, which lists all of the components which are candidate molecules identified from your sample. And if you click on any of these in particular, you get the information shown down below. So in our particular case, we were using the ion mobility QTOF, so we've got our CCS value incorporated as part of this identification tool. We get our component list, which is exportable and queryable in a variety of ways. We can illustrate the chromatogram for each of the components. So as you click through the component summary at the top, you'll see this EIC plot change in each case, demonstrating the structure as well on the page there. And then on the right-hand side, we also see sort of cartoon views of the low energy and high energy spectra that go with each of these features. And we're going to zoom in and look a little bit more closely at this low energy and high energy spectrum view. So we're going to zoom in here. So here is the representation of the low energy and high energy spectra for a compound that we found from this sample set. This is rosamycin. This is not one of the standards that we doped in. This is a de novo natural products identification. So you can see on the top here the low energy scan, and you can see the parent mass up here. And then down below in the high energy spectrum, you see not only the parent mass, but you also see predicted annotations for possible fragments for each, or identities for fragments that are in this high energy spectrum. And in this particular case, these fragments are pretty plausible. So the one highlighted here in orange is the A-glycone of the molecule missing the appendant sugar. And then if we look further down in the trace, we find a corresponding fragment which includes the sugar component, but is absent the A-glycone. All of these fragments are annotated. They're available as a list here. And this high energy fragment list if you are confident about this identification, can be exported to your own internal reference library, which then includes both your the structure and the predicted values, as well as the experimental values which you've derived from this review. So over time, you can build a larger and larger library of validated components from your own sample set, and this greatly improves assignment accuracy as the laboratory matures. So this analysis, of course, is done for all the peaks across the whole spectrum, and so you can also look at component plots, which show you now in a single trace what the key features are. Encouragingly, a number of the natural products which we added as dopants to this sample are correctly identified. So we added actinomycin D here on the right. We added erythromycin G. We added tetracycline, which is down here in the bottom. And then the system correctly identified rosomycin. So rosomycin was not, as I said, it was not one of the ones we added. However, we did validate its presence there by isolation and confirmation by NMR. So we know that this identification is correct. And this makes a very nice test case to show how the Natural Products Atlas can then be used for augmenting discovery workflows in this area. So we're going to take rosomycin as a prototypical example. And we're going to go back to the scientific library page, and in particular to look at this synonyms box. So I'm just going to expand the synonyms box so you can see a little bit more clearly what's in it. So the synonyms box has a couple of key pieces of information. At the top here, this is the DOI for the journal article. So you can copy and paste this into your web browser, and it should take you directly to the article. If your library or your institution has permission for access, of course. And then underneath, we have a URL, which is the MPA URL. So this is the, you can't see the whole URL here, but if you copy and paste this URL, it's the unique URL for the page for that particular compound. So this URL will take you straight back to the atlas, to the specific compound that you're interested in. So if we were to copy and paste this URL for the rosomycin page, it would take us straight back to the rosomycin page of the atlas. Encouragingly, this particular molecule has an external link to GMPS. So if you click the GMPS link, what happens is you get a pop-up. I'll just expand that pop-up a little bit here. So you get a pop-up window in the Atlas page, which lists all of the spectra in GMPS which match to this molecule. So in this particular case, there's only one reference spectrum, but sometimes this includes a much larger list of candidate spectra, and you can choose them based on assignment quality and other factors. So if you follow the link here, so if we click this link here, this takes us then directly to the reference page in the GMPS website. So I'm going to click that now. So as a quick primer for those who maybe haven't heard of GMPS, so GMPS stands for the Global Natural Product Social Molecular Network, and it's a large community-driven project to accumulate experimentally acquired MSMS data for natural products and other biomolecules of interest. So it's a growing repository of spectra contributed by the community. And the GMPS architecture includes quite a lot of tools to allow you to sort of search and cross-check molecules against one another. If you were to follow the link from the Atlas, you would get to this screenshot, which is on the right, which is a screenshot of a part of the rosomycin page in GMPS. So you've got a lot of the same information the Atlas contains, including information about structure and various other things. But importantly, there are also tools like MZSpec, so you can download the MSMS spectrum, you can compare the MSMS spectrum against other spectra in the data set, and so on and so on. So this is a very valuable way to compare data that you've acquired against data acquired by others in the community. Encouragingly, it has reciprocal connections, so you can see the Atlas there listed, and you could follow that, and it will take you back to the Atlas page again. The other thing which is really valuable, if we go back to the Atlas entry for rosomycin, is that we have this cluster ID. So if we follow the cluster ID link, we'll go to the corresponding cluster page for this molecule, and that's displayed here. And you can see rosomycin is the node highlighted in red on the right-hand side. And you can see in this cluster that there's actually 3 groups of molecules in the same general family. So if we look a bit more closely at what differentiates these sets of molecules, so the subgroup on the right-hand side, this is the rosomycin group, and here is the structure of rosomycin, just for reference. The group in the bottom contains molecules such as mycinomycin 1, which have very, very similar structures in many ways. So the core macrolide, so this component here is very similar between mycinomycin and rosomycin. The pendant amino sugar on the left-hand side is the same. But in this subgroup, we now have a second sugar which has been added at the bottom. And that's what differentiates this group on the bottom left here from the group on the top right. The group on the top left is different again. So this is exemplified by algomycin E. So again, the core macrolide is very similar. In this case, the pendant sugar on the bottom right is the same as in the mycinomycin class. But in this case, the sugar on the top left is now different. And so this is a third subgroup. So these molecules are all structurally very similar. They are likely closely related in terms of biosynthesis. And so it's possible to extract the information from the atlas about just this subset and to use those to build custom-made scientific libraries for more targeted analysis. So we do that using the advanced search page. So here is an example of what the advanced search page looks like. We're just going to zoom in on the search box so that it's a little bit easier to see. So this tool includes a drop-down list here, which contains a very large cross-section of different fields available in the atlas. So there are many more choices in the advanced search page than there are on the basic search page. And it's possible to build much more sophisticated queries using this tool as well. So in this particular case, I've only selected the cluster ID and I've made it equal to the ID number for this particular cluster. But you could then choose an AND or an OR feature here, and then you could have a second feature, whatever you choose. And so you can build much more detailed queries here if you want to. So in this particular case, we only care to get all the molecules, the information on all the molecules from this particular cluster. So if we run this search, you get this results box at the bottom, and we're also going to expand that so we can see a little bit more clearly what's in here. So this is actually a large table. You're not seeing all the data presented here, just a subset of it. You can see that on the first page, we see the first 10 of 69 rows. So there are 69 compounds in this cluster, which is consistent with what we saw before. And encouragingly, rosomycin is one of the ones that we see here listed early on. So this results table, if you look at the advanced search page, there is a tab here called Download Results to TSV. So you can export just these compounds selectively from the atlas directly, and you can then convert that exported file into a scientific library in your software tool of choice. And so in this way then, if we go finally back to our workflow again, so hopefully what I've been able to demonstrate is that the atlas provides a complementary tool to existing software platforms for mass spectrometric analysis, and that it will help not only with the initial annotation phase for identifying molecules within unknown sample sets, but also that the tools within the atlas infrastructure allow you to do more sophisticated analyses and to subset and slice and dice the data set in profitable ways. So, for example, you can abstract information about subsets, clusters of molecules which you think are of highest priority. You can convert those into custom scientific libraries, and you can then use those custom scientific libraries with optimized discovery parameters in order to identify families, low intensity analogs, and other subsets and groups of molecular classes of interest. So the last detail then is to tell the audience how to identify or how to access these various tools. So the atlas is available freely online. It's totally open. It's at npatlas.org. And the scientific libraries for the Atlas data set are available as in the correct format for either Progenesis or Unify at the URL which is listed down here below. I'm going to show this slide again in just a second, but before I close, it's important for me to acknowledge both the terrific team in my own laboratory that's helped enormously with this research project, and also all the other international collaborators who've been an integral part of the development of the Atlas program. The folks at the bottom here who provide the research support, which supports our research program, and lastly, of course, everybody here for your attention. So with that, I'd be very happy to answer any questions.
Great, thank you so much for that excellent presentation, Roger. We will now move on to the Q&A, and I'll remind our audience that to ask a question, please enter it into the box on the left-hand side of your screen and click Submit. We've got about 10 minutes for questions, so we're going to see how many we can get through. I'll kick straight off. Here's a question, Roger. For a new user of a natural product atlas using mass spectrometry data, what would be the best way to approach this resource?
Okay, well, thanks very much for hosting the Q&A session here. So I would say that there are two ways that you could approach that. The most straightforward way to use the atlas for new users would be to take individual pieces of data from your mass spectrometry analysis and then search them directly in the basic search page. So the basic search page is the sort of gateway to the atlas. It's the easiest tool to use. It's the most straightforward. It contains quite a lot of fields which are relevant to mass spectrometry. So you can search by accurate mass, you can search by elements of the formula, you can filter the data by taxonomic origin and things like this. And so this will give you feedback about candidate molecules for particular analytes. And you can then use the results page from one analyte to look for other related members and look sort of reciprocally back and forth between the Atlas data set and your own mass spectrometry data set to find other family members and other elements of the same molecular class. So the basic search is undoubtedly the most straightforward place to gain entry to this.
Great, thank you, Roger. A general question here about the Atlas. What are your immediate and longer-term future plans for the Natural Product Atlas?
Well, this is a great question. We get this one quite a lot. So we have, in the immediate term, we are working on making sure that the data set, the microbial data set, is as comprehensive as possible. So we're doing lots of work trying to capture legacy data from the scientific literature, make sure we have all the older molecules and keeping on top of what's coming out now. And we're also making structural changes. So for developers who are in the audience, there are plans in the works to release a fully fledged API version of the Atlas later in the year. So that will allow other external tools to interact with the Atlas more efficiently. And then our long-term dream would be to make this Atlas fully comprehensive, so that it covered molecules from plants and from the marine invertebrate world and things like this. That's obviously a much larger goal, but it would be amazing were we able to build it out to cover a greater swath of biological space as well.
Absolutely, Roger. And this next question kind of falls on from that, because this user said, very true point here, libraries and databases are becoming bigger every day, and these bigger libraries can often lead to more false positives. So do you have any sort of best practice tips or recommendations for the best use of the MP Atlas with this in mind?
Yeah, that's another really good question and a very fair point. So it's quite a difficult question or problem to resolve accurately. One of the other developments that's going on with the Atlas right now is we're trying to improve the sort of taxonomic characterization. So the current version of the Atlas includes information about genus and species, but it doesn't include any of the information about other taxonomic ranks. And that makes it difficult, for example, to select just the cyanobacterial compounds or just the myxobacterial compounds and things like this. And so another development which is coming online in 2020 is an improved taxonomic hierarchy. And that will allow you to filter just for subsets of producing organisms, which is likely to improve the accuracy of assignment quite a lot. The other thing we would like to do is to try and capture all the instances of discovery from the literature rather than just the first one, because that would tell you about how many different kinds of organisms a molecule was found in and how that molecule is distributed. So that's obviously a much larger data curation challenge. We're still thinking about how we might do that. But the taxonomic rank information is well underway, and that should come out later in the year.
Thanks, Roger. A more technical question here. Can I build my own custom library in Unify and search within that custom built library? And if so, what experimental parameters can I include in that custom library?
Yeah, that's a good question. So there are a few ways you can do that. You can do custom searches in the Atlas, and you can export from the advanced search page, as we saw in the end of the presentation there. That export file is a TSV file. It's not in the scientific library format directly. You'd have to convert that, but it's certainly possible to do. You can do it by hand if there aren't too many of them. So that's one possibility. You can also filter the larger scientific library using the tags. So you can take the standard library that you insert, and you can only include optionally certain genera or certain species and things like this. So there are ways to sort of slice and dice the data. In terms of which parameters are used at the moment, so at the moment it includes the accurate mass, the isotopic distribution, and there are plans in the next release to also incorporate theoretical CCS values. So that is not in the current scientific library version, but the next one that comes out should also include this theoretical CCS, which, if you have an iMobility enabled instrument, would obviously add an extra parameter for you to compare.
Thanks so much, Roger. A good question here. Someone's asking just what's the best way to contribute to the Natural Product Atlas effort?
Oh, that's great. We love people who are willing to contribute to the Atlas. So there are really two ways that you can contribute. If you know of molecules which we don't have, then there is a deposit button on every page of the Atlas. The deposit button is in the top right corner. So you can deposit articles which we have not captured yet. It's very easy to do. It takes only a few minutes. We need a very small amount of information. We really only need to know what the article is, what the compounds are, what their names are, and what the organism is. So it only takes a few minutes to do. Alternatively, if you find mistakes or corrections or updates that we haven't captured that we ought to, there is also an additions and corrections section where you can submit suggestions for things that need to be updated. And we have a curation team on hand here in the background who will review all of those. And we do database releases several times a year, and so we will incorporate those corrections and include them in the next release.
Great, thank you, Roger. A question here about the cluster feature. What is the advantage of the cluster feature, and how can the cluster analysis be useful for new identifications?
Yeah, so what the cluster feature does is it identifies all the molecules in the data set which are structurally related to any reference molecule that you've chosen. And this is not done by compound name or by the publication in which they were produced. It's done only based on comparing the chemical structures to one another. And so this can be very helpful at identifying all the members of a family, regardless of when and where they were found and published. And so it can be very helpful for making sure that you understand the sort of landscape of analogs for any class that you're interested in. And you can use that in order to improve your confidence about assignments about particular groups in molecules and things like this.
Great, thank you, Roger. A question here, again, about Unify. How do we use NP Atlas for Unify? On the NP Atlas website or from the Unify software?
So if you're a Unify user, the easiest thing to do is to download the scientific library and install it, because then the Atlas dataset, the complete Atlas dataset, is in Unify in one go. But as we demonstrated at the end there, you can go back and forwards between Unify and the Atlas in order to add value. So you can, as we talked about in a previous question there, you can build these custom queries, and you can build dedicated sub-libraries if you know that you're interested in particular classes or particular types of molecules. You can also do things like use the compound search box to search for substructures and you can run advanced searches which will look for only molecules that have particular motifs. And so there are really lots of ways in which you can build annotation libraries which answer specific questions that you might have about your sample set. So I would say that for general workflows, it's best to download the scientific library and use it directly. For targeted queries, you're best to craft these custom queries and then import them separately.
Great, thank you, Roger. We're running out of time for the Q&A, so I'm going to ask one final question. I'd just like to say that we've had a really, really great response to their questions, and we're going to do our best to get you responses to your question offline after the webinar is finished. This question comes from the University of Sao Paulo. We've had a really international question base today from Oklahoma, from Strathclyde, from New York, and this one's from Sao Paulo. This questioner says, thank you for the great talk. Do you intend to include natural products from marine macroorganisms into the NP Atlas?
Yeah, this is so this came up a little bit earlier in the Q&A. So absolutely, we would love to extend the Atlas to cover more of the natural world. It's quite a big challenge. So the Atlas currently contains about 25,000 entries. It's very hard to predict how many microbial natural products have ever been published, but perhaps 40,000, something like that. The marine world is of a roughly similar size, so there may be as many as another 40,000 to find there. And then the plant world is an order of magnitude larger again. So the plant world is well over 200,000, maybe quite a bit more than that. So the scale of effort required to extend to these other two sources is quite high, but we're working with funding agencies and other collaborators around the world in order to try and do that. So if the audience members are enthusiastic about sort of helping us to extend the scope of the Atlas, you can send us an email, support@npatlas.org, and let us know how you might help or give us ideas about how we might work to expand that. We would love to do that very much. So it's a great suggestion.
Great, thank you, Roger. The task is pretty mammoth, but it's good to know that everyone's collaborating and lending a hand. So thank you very much for your time today, and I'm sure our listeners really enjoyed your talk just as much as I did, and I'd like to give them a big thank for attending today as well. That's all the time we have for questions, and yeah, thank you to everyone for attending, and once again, thank you to Roger for his talk. Goodbye.
My pleasure. Thank you very much.